Hello and welcome to my genetics lab. Today we will be discussing transcription in prokaryotes. The stages of transcription in order are template binding, chain initiation, chain elongation, and chain termination. First, we need our two DNA strands. This top strand is going from 5' to 3' and the bottom goes from 3' to 5'. With transcription going from left to right, we will add in our RNA going from 5' to 3' on the bottom strand because RNA is transcribed 5' to 3'. Nucleotides are added to the 3' end of our RNA. The bottom strand is our template strand because it is complementary to RNA. The top strand is our coding strand because it has the exact sequence of our RNA. The holoenzyme has sigma, which helps with binding and allows initiation to begin transcription. The nucleotide here is denoted as +1 because this is the start of transcription where our first nucleotide of our gene is transcribed. Here is our promoter region where RNA polymerase binds and begins transcription. This -10 region is where we have our consensus sequence where RNA polymerase binds. This region is also known as the Tata box. The -35 sequence is also important for RNA polymerase binding. The upstream region has our negative values and the downstream region has our positive values. Downstream is where we have our RNA coding region that is continually transcribed until it reaches the terminator region. I will now show an example of an inverted repeat to demonstrate a possible type of termination that is row independent. We see our DNA being transcribed to RNA, but in an inverted repeat, the bottom strand will flip to look like this. The core enzyme without sigma is involved in elongation and it helps with termination. So let's say we have these specific codons for our DNA and RNA. This inverted repeat will cause a termination hairpin loop that looks like this. We get this poly A tail on the DNA and poly U tail on the RNA, which signifies termination. This binding is weak and allows for the RNA to release from RNA polymerase and terminate transcription. Now, let's say we are creating a gene that codes for an enzyme to metabolize sugar and the -10 box is deleted. The sigma subunit would not recognize this -10 consensus, or Tata box, so the holoenzyme would not be able to help bind and allow transcription to begin. So the gene for this enzyme would not function correctly to metabolize sugar.